To solve problems like this, a discipline called chemical biology emerged. Chemical biology fosters interactions between the fields of biology, chemistry, and physics. This is the story of a network of chemical biologists who came together to create the first universal tool to image and measure plasma membrane tension in action. But how did the researchers imagine what this tool would even look like? Their first instinct was to turn to what already exists in nature for inspiration.
Actually, we thought maybe we could do it just the way lobsters do it, the way lobster change color during cooking. Because actually the pigment in the shell of lobster, it's a carotenoid, it has the orange color of carrots, but in living lobster, it is kind of flattened and in a way stretched by forces from the surrounding protein until it really kind of literally turns blue. And so only during cooking then the protein is melted away, the pigment is released and relaxes into the natural form and produces the orange color.
So we decided to adopt this idea and created fluorescent molecules that behave similarly inside membranes. This lobster-inspired molecular probe was able to insert into simple artificial membranes where its visual properties were modified depending on the strength of the surrounding forces, such tension. However, it wasn't bright nor sensitive enough for application in real cells, where changes in tension are very small.
The scientists needed another idea. And once more, inspiration came from outside the lab.
So one day I arrived at the group meeting with a picture of a scuba diver. And these huge shiny flippers were for me the perfect image of the probe that I had in mind, which had stronger fluorescence and a larger surface to better feel the forces in the environment. My design had two molecular flippers, which were able to turn relative to each other depending on the forces they were experiencing. And this movement is what in turn determines the visual properties of the object.
Marta's flipper probe worked beautifully in artificial membranes, showing bright fluorescence and obvious changes depending on the level of surrounding forces. However, Marta's probe behaved horribly in cells. The emotional roller coaster wasn't over for the scientists, who had yet another problem to solve.
After many attempts, I finally discovered the perfect structure that ensures that the flipper probe would remain inside the plasma membrane and not be toxic for the cells.
Scientists just needed to add a few drops of the probe mixture to the cell culture before observing it under specific lighting conditions. Now, depending on the level of tension, the plasma membrane appeared in different colors.
It took me another 2 long years to calibrate the flipper probe in different cell types and make sure we understood how it was working.
But this wasn't the end of the flipper saga. The content of cells are also organized into smaller compartments separated by similar membranes. These compartments include mitochondria, responsible for energy production, the endoplasmic reticulum, crucial for protein production, and lysosomes, which can be compared to the recycling plants of the cell. The intracellular membrane surrounding these compartments and the forces that control their function were, so far, completely beyond the reach of scientists.
Tweaking the structure of the flipper probe led to a full repertoire of fluorescent molecules. Each of them is able to target the membrane of one specific compartment and report on its level of tension in a similar manner as for the plasma membrane.
It's really exciting. For the first time, we are able to observe the consequences of an invisible force acting in real time inside the cells.
This breakthrough would not have been possible without the close collaboration of scientists with complementary sets of knowledge and skills, brought together by chemical biology. It is proof that the future of science lies in our capacity to break down barriers between traditional disciplines. Physical forces cannot be seen, but are what sets the world in motion all around us. Before today, had you ever imagined that similar forces were constantly at work within your body? Or within each of your cells? Membrane tension is one of these invisible forces that needed to be measured in order to better understand its role in living cells. 10 years after the initial idea, the Flipper probes developed by chemical biologists are now used worldwide to visualize and measure membrane tension inside cells. This new tool opens a brand new door into the study of life sciences, and potentially into the development of concrete new treatments for many diseases.
So I hope you enjoyed the video. That's certainly something that we're very proud of as a group, to be able to have created such a set of tools that allow people for the first time to measure something that beforehand was really very difficult, if not impossible, to measure. And indeed, in my own lab, we've seen that if you manipulate membrane tension, this has very strong effects on our TOR signaling pathway. So that before that, we always thought TOR signaling was regulated only by nutrients, but this demonstrated that not only nutrients regulate TOR, but also biophysical properties of the cell. So this opened a whole new research avenue for my group. Okay, so I think I've summarized for you the types of things that are possible within this master's program, the types of projects. The global aim is to do interdisciplinary research. We are looking for very talented students. We're very selective. So you should, every student that's accepted into the program goes through an interview process. You should have a strong background in biology, chemistry, biochemistry, biophysics, or equivalent, and certainly be prepared to be put outside of your comfort zone. So we do this by design to try and make the students more well-rounded by the time that they finish their master's degree. The starting date for the next intake is September 2023. If you need a visa to live in Switzerland, then the application deadline is February 28th. Formally for the university, others need to apply by April 30th. But actually we have an internal deadline because we do this selection process, and this internal deadline for us would be March 15th. And we have a website that you can apply to and fill out information, and this will allow us then to discuss with you and make sure that you would be a right fit for the program, and also that the program would be the right fit for you. So the fees for the program are relatively modest, certainly compared to North American fees. We do also try to give our students a stipend. So this was easier when we were still part of the NCCR. Now it becomes a bit more challenging, but for the next year or two, we still have money in place that we can pay a stipend. But we're very enthused by students that can get their own fellowship, for example, with this excellence fellowship program. So this is based solely on excellence and is attainable by students that have really done well in their undergraduate studies. And so we would certainly very strongly encourage all applicants to our program to apply to this Excellence Fellowship Program to help secure funding. That more or less covers everything I wanted to share with you.